I apologize in advance if any of the science seems slightly simplified or the knowledge repetitive. I'm sending this video to my family as well, and none of them have spent the last 2 months in a lab dedicated to skin research. My project is intended mostly to study melanoma tumors in mouse lungs. Melanoma is a particularly aggressive form of cancer. Of the 7 most common cancers in the US, melanoma is the only one whose incidence is increasing. Someone dies of melanoma every hour. This project will observe how melanoma adapts once it is targeted by the immune system. Growing the melanoma was pretty neat. It actually dyed the media it was in black because of all the pigment it was producing. It was very easy to tell which plates were thriving. Ironically though, it's the ability of the cells to show light that allows you to see them in the lungs of the mouse. These cells contain luciferase, which is an enzyme that is also found in fireflies. When a chemical called luciferin is injected into the mice, luciferase cleaves it, and this produces light, which can be picked up by imaging machines so you can actually see the melanoma while it's in the lungs. Of course, first I needed to get the melanoma into the lungs. You'd think one of the perks of studying skin cancer is you wouldn't have to worry about cancer in the internal organs. As the picture on the left indicates, if you can inject something into the tail vein, it will go almost directly to the lungs. The contraption on the right is how we hold the mice still enough to inject them. I mean, they're not very thrilled about the process, and they have very tiny, hard-to-see veins. After the melanoma is injected, it takes about a week or 2 for an actual visible tumor to form. On July 10th, I injected several mice with melanoma. These are the remaining 3 that were imaged August 5th. As you can see, after more than a couple weeks went by, they had a lot of cancer in their lungs. The one on the far right also has melanoma in its tail, probably due to a few missed injections before I actually got the vein. These are 2 wild-type females I injected with melanoma July 30th. As you can see, they haven't progressed quite as far as the other ones who had a much greater head start, but the one on the left does clearly have melanoma in its lungs. The mouse on the left was also vaccinated against melanoma, so in the coming weeks when the mice are imaged again, we should see the tumor progressing slightly, but then as the vaccine takes effect, it should begin to decrease, and then as the vaccine loses effect, it should start increasing again, while the mouse on the right should have melanoma appear in the lungs and then just continue to grow. It'll be a nice control. These are some wild-type males I injected with melanoma on July 30th. 2 of these mice were vaccinated against melanoma like the female I mentioned in the last slide. I hope to see a similar tumor progression in all the vaccinated mice, with the tumor growing again once the vaccine loses its effectiveness. We can then examine the cells of these tumors to try to understand why these cells have evaded the immune response. I don't have images for the remaining mice because they were injected August 7th. They're a wild-type male and several immunocompromised mice, which should serve to highlight the difference in tumor progression in mice that have adaptive immunity, like the ones that were vaccinated. It is a challenge to find peptides specific to melanoma cells. Directing your immune system to attack something that is also found in non-cancer cells could produce an autoimmune response. In fact, vitiligo, a condition where normal melanocytes are destroyed, sometimes correlates with response to melanoma treatment. The woman above developed vitiligo after undergoing immunotherapy for melanoma. But you can also err on the side of caution and pick a peptide that may not be in every tumor cell, and then part of the tumor could survive. Melanoma antigen recognized by T cells, or MART1, was the main ingredient of the vaccine. MART1 has already been used in peptide form in various clinical trials and have shown promising results. In phase 1 trials, it has been used in combination with other peptides in dendritic cells as a method of inducing an immune response. In the mice, I expect to see the tumors shrink as cells containing MART1 are targeted, but eventually, due to something unknown, such as downregulation of MART1 and perhaps upregulation of some other peptide, or the T cells being exposed to cytotoxic T lymphocyte antigen 4 or some other molecule that regulates. Once the tumors have undergone a process to avoid being destroyed by the vaccine, we can sequence the cells and determine the DNA modifications that might have occurred, if there are any, and try to develop new therapies that would target these modified cells and hopefully have something that's more effective against melanoma. Scientists have currently identified a lot of peptides that correlate with melanoma. I hope my project will contribute to the understanding of how these peptides can be used against melanoma. And last but not least, I'd like to say thank you to Dr. Roop for allowing me to be here. This was such an amazing summer. And thank you, Brad, for showing me how to do like everything from pipetting to how to handle mice. Thank you everyone at the lab for making this such an incredible summer. I had so much fun and I learned so much. So, any questions? Just kidding.